Hello, my name is Ernie Guzman. I'm the Senior Manager of Technical Support at Twist Bioscience, and I'd like to welcome all of you to today's presentation entitled Comprehensive Identification of Multiple Respiratory Viruses, including SARS-CoV-2 in a Single Assay. It looks like we have a number of people joining us today from Kenya, Buenos Aires, Portugal. That's fantastic. We'd like to extend a warm welcome to all of you. Today, we have two speakers who will talk about how the COVID-19 pandemic has highlighted the urgent need for genomic tools that can accurately and fully characterize samples, allowing for the identification of pathogens that present with varied, but often very similar symptoms. Just a couple of housekeeping rules to cover. First, all lines will be muted during the webinar. This is to reduce any distractions during the presentations. Secondly, we will have a Q&A session at the end. Please submit your questions in the Q&A box. We will answer your questions after the presentations are complete. And finally, following the presentations, there will be a short 1-minute survey that we would really appreciate you taking. Your feedback will help us improve future webinars. Our featured speaker today is Nick Greenfield. Nick is the founder and CEO of One Codex, a microbiome data analysis platform based in San Francisco. One Codex works with a broad range of leading academic, commercial, and clinical institutions. Its software and database have won awards and top challenges, including CDC's No Petri Dish Challenge and the FDA's Pathogen Detection Challenge. Prior to One Codex, Nick worked as a software engineer, data scientist, and strategy consultant on a range of scientific computing, machine learning, and economic modeling problems. Nick holds a BA in International Relations and an MA in Environmental Studies from Brown University. But first, we will hear from our very own Venkatesh Moktali. Venkatesh is a senior product manager at Twist Bioscience. Prior to joining Twist, Venkatesh has been instrumental in launching several key products in the genomics industry, with stints at Thermo Fisher Scientific and Mission Bio. Venkatesh holds a PhD in bioinformatics and genomics from Penn State University and a BS in bioinformatics from the Vellore Institute of Technology in India. And with that, I'd like to hand it over to Venkatesh.
Thank you, Ernie, for the kind introduction. Can you hear me well, Tim, on the line, or Ernie?
Yes, we're all set.
Great. Okay. So good morning and afternoon to everyone joining us today. I know we have quite a few countries as well as regions represented, so that's really exciting. I'm very excited to be speaking to you about our latest NGS panel product. Just a quick note, today we will have a few poll questions during the presentation, one at the beginning, one in the middle, and then one towards the end. So essentially these poll questions help us in understanding your needs, our customer needs better. And so I really appreciate your responses. So before I start with my presentation, I wanted to request you to provide your response to the first poll question that's going to come up on your screen. Perfect. We are in excess of 70%, and it looks like most of the audience today is doing either research or diagnostic development. Again, thanks so much for your responses. And with that, I'll start with my presentation today. So perfect. So I'll begin my presentation today with some background on respiratory viruses and respiratory infections. What you're seeing on screen is a bunch of viruses that infect upper and lower respiratory tracts. Overall, globally, there are about 4 million deaths that are caused by respiratory infections. Half of these are due to viruses, which tells me a massive figure, right? And depending on the individual's health and immunity, even common viruses can cause serious respiratory infections. These respiratory infections can be, as I mentioned earlier, categorized into those infecting the upper respiratory tract and those that infect the lower respiratory tract. So, for example, the coronaviruses are known to infect the upper respiratory tract, while influenza infects the lower respiratory tract. Now, in terms of when I list out these respiratory viruses, the second question that comes to mind is, okay, why does it matter? Why do we need to be able to differentiate between different viruses? The reason being that a lot of these viruses share initial symptoms and are highly similar in the symptoms that patients display. And so some of these patients will quickly deteriorate, while others can go back home and recover easily. So it is critical to understand the causative virus or viruses in helping with appropriate isolation practices. What you're seeing on the left-hand side of your screen is a table from a study that was published in 2017. The study looked at different causative agents of community-acquired pneumonia. Essentially, they looked at individuals that came to the hospital reporting symptoms of a community-acquired pneumonia, and then they looked at what were actually the causative agents. And so, what you're seeing on the in the table in the leftmost column are the virus types that were identified from each of these infections. And so what you'll see is that even though all of these individuals had community acquired pneumonia, you will see that there were a number of different viruses who were causing these symptoms. And so what that tells us is that the symptoms may be exactly identical, but in order to make sure that we are able to provide the right kind of isolation practices and therapies, we need to be able to identify what is the causative agent. And not just separating out different viral species, right? Even separating viral from bacterial causes can help us avoid unnecessary medication, and as well as then choosing specific antiviral agents based on these identified virus. And that's exactly where our panel comes in. So our respiratory virus NGS target enrichment panel is able to not only just identify a virus, but it is able to identify co-infections as well. In addition, you can identify virus more quickly even when there isn't a prevalent seasonal virus. And that is a great advantage when you compare them with conventional assays, such as the immunosorbent assays or PCR assays that are highly dependent on previous knowledge of viruses. And so oftentimes they are ineffective in identifying new pathogens. And in addition, rare viruses can not be detected using these conventional methods. And that's why our NGST panel really comes out with these advantages. With that, I'd like to maybe let's switch gears and start talking about the panel more. I want to start with giving you a glimpse of the content on this panel. So our respiratory virus research panel covers viruses from all the major respiratory viral clades. So as you can see on screen, we have a picture, a representation of the taxonomic tree, and you'll see that we cover virus from each of these major viral clades. And so when we were designing this panel, we made a conscious effort to cover as much respiratory viral diversity as we could while keeping the design size compact. And in essence, in order to cover each of these reference genomes, we have tiled them 4x, and so as to cover have a comprehensive coverage of each of these reference genomes from each of these representative viruses. And then when you look at the probes that target each of these viruses, you'll see that we have, out of a total of 41,000 probes, the reference sequences contribute about 18,000 probes. And then what we've done is we've added additional probes to cover this massive diversity that we've seen in influenza and rhinovirus types. So almost about 22,000 probes combined that cover this additional diversity in these two specific clades. And so ultimately what we have is an extremely robust panel design that covers this incredible viral diversity. And so you should be able to always get an identification within this respiratory viral group. And so that was our rationale behind the panel design. Now, before I dive into some more details into panel performance, we have one more quick poll question for you. So let me quickly ask for your attention on the upcoming poll. At 62%, perfect. Okay, so we'll stop there. It looks like a majority of the folks online today are interested in viral detection and co-infection. Fantastic. Thanks again for sharing your opinion through these polls. All right, let me get back to let me get back to the presentation. So let's spend a little bit of the next about 15 minutes talking about the panel performance. What you're seeing on screen is some of the data that we generated from the panel. We started with spiking in synthetic controls that we released along with the panel into 50 nanograms of human carrier RNA at about 1 million copies prior to cDNA synthesis. And we then we saw that at about 1 million sequence reads, all templates were covered at around 1500x of median depth. And that essentially covers at least 99% of bases at 30x depth. So what you'll see at these metrics, at this particular experiment setup, is that we see great performance across the different species that are covered on this panel. So with our synthetic controls, we have about 15 different synthetic controls. So we tested against each of these synthetic controls. And as you'll see in the image on the left-hand side, we are able to get best-in-class performance across different metrics. And we looked at coverage, we looked at on-target rate, and we also looked at fold enrichment. And what you'll see is we get massive fold enrichment from each of these individuals, and this enables us to go as low as even 100 copies. And so again, that comes through from the fold enrichment that you'll see in this panel, as well as at the same time getting that fold enrichment with with a consistently high percent on target reads and great uniformity. So to start you off with, again, we looked at these performance metrics with our synthetic controls. We also looked at potential for detecting co-infections. And so in an experiment to look at co-infections, we tested mixing two different synthetic controls into the same human RNA background, and we looked at a couple replicates of these spike-ins. And what we saw was that irrespective of different combinations, so what you're seeing on screen is we combined SARS-CoV-2 with rhinoviruses, we combined SARS-CoV-2 with influenza, and as well as the Boca virus, which is the smallest of the viruses that we tested against. And you'll see that across these different combinations, consistently we are able to detect these co-infections. And you can see that through the fold enrichment numbers that are on screen in the table. So if you look at on the left-hand side, it shows the fold enrichment numbers for strain 1, and on the right-hand side are fold enrichment numbers for strain 2. And so even for a virus that is super small, such as Boca virus, you'll still see great coverage even at 30x. And so that goes to show the performance of our panel. And so again, this is just a representative. Out in the field, you will see a much bigger diversity. And of course, our goal was to be able to catch even the smallest of viral templates, and as well as much larger viral templates, which are similar to the SARS-CoV-2 that we've observed. Talking a little bit about the panel performance in terms of limit of detection. So we also ran a similar experiment with our synthetic controls spiked into human background, where we looked at copy numbers starting from 1 million all the way down to just 100 copies spiked into the human background. And of course, what we wanted to do is we wanted to look at detection of the virus at these different spike in concentrations. And what you're seeing on screen is a performance of the panel throughout these different if you will, CT values that different clinicians will observe while testing these samples. And what you'll see is that irrespective of these spike-ins, even at as low as 100 copies, we are still able to detect these viruses. So what you now are able to see is that the panel not only provides you with great ability to identify co-infections, but also identify those really difficult to work with samples that have low viral copy number. All right, so talking a little bit about coverage. So we, again, in a continuity of experiments, we tested for different metrics. Even with coverage, we looked at different titers through different strains, and we wanted to measure how the coverage varies when you lower the amount of virus that is available in the sample. And what you're seeing in the pilot plot on the right-hand side is that if you look through replicates, you see consistently good coverage of the viral genome, even when you go down to only 100 copies. It's only in a particular replicate that we actually start seeing a few variations, which you see in that top row on the right-hand side image. But otherwise, you will see that you consistently get good coverage even at very low viral copy numbers. All right. Again, I'll keep continuing in talking more about some of the value props here. Even we know that our customers multiplex, especially with high throughput labs as well as labs that get a lot of samples. We tested the panel performance in singleplex as well as in multiplex, and we essentially pulled 8 samples into a single tube. And what you see is that irrespective of whether you're running in singleplex or multiplex, the performance remains unaffected. And so and the second piece that we also tried, in addition to looking at just the multiplexing ability, we also looked at how the on-target viral reads, if at all they vary when you change the amount of virus available in the sample. And what you'll see in the image on the left-hand side bottom is that when you look at single and multiplex modes across different viral titers going from 100 to 1,000,000 copies, you see consistently good results. So again, trying to replicate some real conditions that customers will observe, and proving that the panel performance remains unchanged and provides superior performance across these different scenarios. Perfect. Now, let me move quickly into talking a little bit more about the protocol itself. So the protocol is quite similar to our SARS-CoV-2 panel that we released earlier this year, in that it is you can perform cDNA synthesis and library preparation in 5 simple steps, and it's a 5-hour turnaround time. Starts with the dilution of the nucleic acid samples, goes through cDNA synthesis, DNA fragmentation, and then ligation, as well as then amplification. And so you can start with any nucleic acid type. If you're starting from RNA, then it generates double-stranded cDNA, and if you're starting from single-stranded DNA, which is often the case, it then converts it into a double-stranded DNA. So irrespective of nucleic acid type, you can still follow the protocol and achieve enrichment through this panel. Perfect. So quickly jumping into talking a little bit more about how do we differentiate from competitive panels. We definitely have seen some competitive products out there with ability to look at respiratory viruses. What I'm showing to you on the left-hand screen is comparing our panel with a competitive product. What you'll see is that the Twist respiratory panel provides much more diversity of viral strains compared to the competition. And so what you're seeing in red blocks are the species that are missing in a particular panel. And you'll see that we've added a lot more diversity. So to give you an example, with influenza and the rhinovirus strains, you're looking at almost 13,500 strains represented in influenza A, and about 700 different sequences in human rhinovirus. And this level of diversity is completely missed in the competitive panel. Now, also because we use longer probes, 120 MERS, we are able to capture far more diversity when you compare that with with shorter probes that are just going to miss out the mismatches that you'll see, and they won't tolerate these kinds of variations. So again, two different pieces where we differentiate. One is just the diversity of the viral strains, and as well as just catching all of this novelty that you will see in these evolving viruses. And those are the two pieces that we are able to catch really well. All right, so I'm coming towards the end of my presentation. I'll want to quickly talk about how we are bundling this product. We are bundling the product today with the software, and the software comes from One Codex, which we'll talk about in a moment. If you look at the workflow steps overall, just trying to give you a sense of what are the parts that we expect users and customers to cover, which is DNA, RNA extraction, and cDNA synthesis are products that will be available from other vendors. We'll provide from Twist and the library product, including enrichment reagents and indexes and the panel itself. And then on the other hand, of course, we are collaborating with One Codex in providing you the software solution. So in terms of the software solution itself, of course, Nick will dive into a lot more detail, but what I wanted to give you quickly is we're partnering with One Codex. It provides great reporting functionality as well as visualizations, and it comes with the panel as part of the complete solution. So today, when you purchase these SKUs, these come in 3 different flavors, 2, 12, and 96 reactions, and they are bundled with the One Codex software. And Nick will now talk a bit more about this software solution. So with that, I will end my talk and I'll hand it over to you, Nick. Thank you.
Great. Thanks, Venki. Let me just start my screen share here. There we go. Thanks so much, Venki, and thank you, Ernie and Tim and Kathleen, for organizing and inviting me to share a little bit today. So, as Venki mentioned, I think one thing we're really excited about with this panel is the fact that we're packaging and providing the analysis alongside. And so I'm going to provide a little bit of brief introduction into what One Codex is and who we are, and then the actual workflow for the analysis of data that's produced from the Twist Respiratory Research Panel, and I'll give you a live demo at the end. So in terms of brief background about One Codex, so we're a sequence to answer data platform for analyzing NGS data with a specific focus on microbiology and virology data. And we've been around for the past 6 years. We're headquartered in San Francisco. That may reveal a bias that we're really a software organization that's built tools for microbiologists, virologists, folks in microbiome research, et cetera, to work with NGS data from microbial samples. And a key part of what we provide folks is the ability to do their analysis in a unified environment, solve certain challenges around reproducibility, solve certain challenges around ease of use. It's a web-based platform. I'll give a demonstration. It's quite intuitive, and then solve problems also around compliance and thinking about, the security and management of data that are more relevant, perhaps as you transition into a clinical environment or into, other types of research environments that require additional guarantees there. And we support both whole genome data, so shotgun sequencing or shotgun metagenomics data. We support amplicon data, commonly in microbiome. You'll see folks working with 16S or 18S or other kind of targeted sequencing data, and then of course capture data. And so with Twist specifically, we've partnered to provide an analysis that's kind of one click and very easy to use to get meaningful reporting in a standardized, consistent way from the Twist Respiratory Virus Research Panel. And at One Codex, we work with a kind of range of different folks across microbiome therapeutics development, across infectious disease, across public health, et cetera. So we've seen kind of lots of these different use cases and can help in other areas as well. So to provide a little bit of background on the Respiratory Virus Research Panel and what it looks like in our system, Venki showed a quick screenshot. We'll show a live example in a minute. But basically, the workflow is such that we've built an analysis that's easy to use. It's one click. It gets your PDF report if you want it. It gets you rich structured data that you can export programmatically via an API all the way at the other end of the technical spectrum if that's what you'd like. But it solves some of these problems around having something that's ready to go out of the box. You don't need to spend cycles developing your pipeline to analyze this data type. You don't need to think about some of the nuances with regards to multi-mapping across viruses and some of the other complexity that do arise when you look at this type of target capture data, et cetera. And so, it's built to detect all the distinct viral pathogens that are present or that are detectable with this panel. And it's included with the kit. So when you purchase from Twist, oh, my slides are out of order. I'll describe a little bit about how it actually works. So the way the actual analysis works is you upload your data to us. It's a raw FASTQ file. So we take it basically right after demultiplexing off the sequencing instrument. And then you'll run the Twist respiratory virus research report on our system, and I'll show how that works. And it's, again, quite simple. And under the hood, we're doing local alignment using Minimap2 against a hybrid or combined index of all these different viruses. We're making sure that we resolve multi-mapping and kind of near equivalent alignments in a way that makes sense for this particular problem domain. And so we basically do in parallel alignment to all the pathogens that the panel can detect. And then we generate kind of a unified report. And that includes, coverage maps and some kind of easy to use tooling where you can really eyeball this thing quickly and understand, okay, I've detected this pathogen in the sample, or I haven't, or there's a little bit of probable evidence. I need to dig into this a little bit further, as well as comprehensively kind of what's been detected or not detected with the panel. And in terms of how this actually works, you purchase the panel from Twist, you'll get a getting started email from us around the time that your materials ship out from Twist. And that'll provide some really simple instructions. We'll actually create an account for you that you can just set a password for. So it's very simple. It includes a link to a getting started guide, as well as a link to some more technical documentation on the analysis and how that works. And then you'll actually, once you produce data, upload a FASTQ file, click a run button on a particular analysis, and then get your results. So to kind of make that all a bit more concrete, what I'm going to do now is share a live demo of this system. Oops, wrong direction. Of our platform and how to analyze data from the respiratory virus panel. So as I mentioned, web-based platform, www.onecodex.com. In the top right, there's a login button. So what I'm going to do here is log into a little demonstration account that we've put together. And this works on Chrome and Firefox and Safari, and you should be able to work from virtually anywhere in the world and access your data and do this type of analysis. So it's very portable. And so what I'm doing here is logging into an account that we've pre-populated with a little bit of demonstration data from the validation work around the respiratory virus panel. And if you were logging in for the first time, you get a helpful message here instead that explained how to get started, how to upload some data. But basically what you see here is each of these cards, if you will, is an individual sample. And for us, a sample means basically a demultiplexed FASTQ file. So there should be typically a one-to-one correspondence to your physical specimen. And you can do all of the obvious things that you kind of want in terms of organizing that data. So our system will actually kind of detect what sequencing instrument it's from. It will make some other imputations about your data. You can tag things, you can add structured metadata, you can search, organize. We have folks with thousands of samples in these accounts instead of just 5, where some of that functionality becomes a lot more important and a lot more helpful. And in terms of how you would get started, there are a variety of ways of getting data into the platform, but kind of the simplest is basically my Zoom controls, there we go. The simplest is kind of a drag and drop upload. So if you had a FASTQ file on your desktop or on another drive that you had access to, you can basically drag and drop your FASTQ file into the browser. It's uploading here. This is 1 million reads. So a kind of realistic depth that you might see for this type of data, and drag and drop. We also, you can upload your data using a command line tool that we have. You can import data from base space if you're pushing data to base space. So a variety of ways of getting things into the platform. And then if we go back over here to this samples tab, this is the file that we just uploaded. You can see that we're kind of starting the analysis in the background. We'll come back to this particular sample towards the end of this demo and look at the results. Again, we can do all the things you expect. Demo on August, whatever the date is, 26th. So to look at a few of these examples more concretely, this is the respiratory virus panel report for a synthetic sample that's kind of a mix of both the rhinovirus and SARS-CoV-2. And what you can see here is kind of a standardized report that says, we're detecting human rhinovirus 89, SARS-CoV-2. There's a useful and kind of quick visual snapshot of what that means in this case, right? So we're detecting 100%. We have 100% coverage of both viruses, 220x depth on one, 720x depth on the other, high identity. A note about these is these are the raw identities. So this is, in part, you'd always expect something a little bit less than 100 just due to sequencing error. But both of these are definitively present in the sample. We have a little bit of mapping to the SARS genome here as well, that we're calling as an indeterminate result. And then we have kind of an exhaustive report of everything in the panel and whether it's detected or not detected present here. So this is what a mixed sample looks like. A simpler kind of single pathogen example would be this report where we have human parainfluenza virus 1, and we basically get a really clean detection of a single result, 100x coverage, very, very high depth. Again, this high kind of 99% plus identity, a little bit of information and link out on the methodology, and then, kind of more comprehensive results reporting as an appendix here. And this all sits on top of our platform, where there's kind of standardized analysis. So this is analyzing in the background. To actually run the twist panel, what we do here is grab the sample that we uploaded at the beginning of this demo, and we'd scroll down here, and this is all included in that getting started email, to the twist respiratory virus report, and we'd click run. And for a sample of this size, 1 million reads, depending on how my live demo is going, that takes, on the order of 2 or 3 minutes. So we should see results. What's happened here is kind of the first set of results have passed. This is our metagenomic analysis against the One Codex database, which is kind of our standard database. We see all the reads mapped to human respiratory virus 1, and the file that I'm showing is the same file as that one that I just showed before. And we kind of present, a visual representation of the taxonomic mapping, an overview, and some information about how reads map within the sample. Again, you can see the vast majority of reads in the sample map directly to human respiratory virus 1, and we estimate some depth and some other information. And then in the background, that report that I just mentioned is also, that kind of clean visual PDF is also rendering and generating, and that should finish in the next minute or so. And I think to provide a couple other examples of what some of these reports can look like, here's a flu SARS-CoV-2, again, a mixed sample where we can kind of cleanly detect the presence of both viruses, a little bit of off-target mapping, again, kind of comprehensive results. And for SARS-CoV-2, we'll also kick off some other analysis that includes SNPs and calls SNPs against the original Wuhan strain, should those be present, that lets you download a consensus sequence or consensus genome, again, against the original Wuhan strain, MN908947, and could be taken and published into, pushed to NCBI or pushed to GSAID or other repositories, so that it can be used for kind of epi purposes. And I think a big plus of how we're doing this analysis and a big value of doing this, obviously, with sequencing over, a different type of multiplex molecular diagnostic, is, of course, the ability to cover these genomes, in their entirety or close to their entirety, and to recover SNPs in a meaningful way out of these genomes so that we can start to look at questions of lineage, start to look at questions of, how outbreaks might be unfolding, plug things in along those lines. So at this point, I think the sample that we ran a little bit earlier, it still says pending, should be almost finished. So we'll give that another 30 seconds or so. And then we'll look at that report that we uploaded and kind of get things started from there. And again, all of this is documented on our site, and this is included in the emails. There's kind of introductory documentation around how to get started with our platform, and then critically, how to get started specifically with the Twist Respiratory Virus Research Panel. So this is all included when you get the kit. We'll send you an email. It includes your kind of step-by-step getting started instructions, how to upload your data, how to analyze your data, and what those results can typically look like. So that's kind of the main thing that I wanted to walk through here, is, how, when you purchase the Twist Respiratory Virus Research Panel, the bundled analysis works, which is that we'll send you an email as the materials ship, and that'll include us helping to create an account for you. You'll sign up, simply set a password. Your account will be credited so that you can do kind of the analysis for the number of reactions that you've purchased with the Twist kit. Process of uploading FASTQ data to our site, just again, simply kind of drag and drop or import from BaseSpace or upload via a command line tool that can be installed on any server. How kind of a default analysis progresses, how to run the Twist respiratory virus report, and then again, what those respiratory virus report results look like, and what a mixed infection looks like, as well as an individual kind of targeting example. So those are, and now it's finished, of course, for the end of my webinar. So these are the results that we uploaded about 7 minutes ago. And again, 1 million reads, we're mapping to all 29 targets within the panel. And here we again detect human parainfluenza virus 1, 100% coverage of the genome, 4,000-fold depth approximately, high identity, kind of definitive detection, and then the more comprehensive results. So with that, I want to open up for any questions for myself and for Venki and others on the Twist side. And it's been kind of our pleasure to walk through the panel and both how it was designed, how it works on the bench side, as well as kind of the dry side analysis that we're providing and packaging with the kit.
Okay, well, I'd like to thank both of our speakers, Nick and Venkatesh. There are a couple of questions from the audience. So for Venkatesh, for the 1x and 30x performance testing, were only synthetics tested, or did you extract nucleic acids from pooled or heat inactivated virus particles? And then a second, on top of that, when you use synthetic, were the synthetics full length or just portions of the viral genome?
Yeah, great question. So we did test against synthetic viruses that were full length sequences for the entire viral genome. We have not tested any extracted nucleic acids, but we have worked with a bunch of customers since since releasing the panel, as well as while we were releasing the panel as part of our early access. And what we've seen is that customers have used different sample types and are able to get enrichment that actually is as good as what we see with the synthetics. So yeah, it should work well in either way. Yeah.
Okay. For Nick, is the One Codex user adjustable? For example, if I want to spike some more probes into the respiratory panel?
Yeah, so the specific report that I showed, the PDF report, is against those 29 targets covered by the respiratory virus. But I also showed for that initial upload kind of a result against the One Codex database. And that's a database of about 150,000 distinct genomes, largely bacteria, but 30 or 40,000 viral genomes as well. And so that works against untargeted data, shotgun metagenomics data and the like, and can be used if, for example, you had additional spike-ins and additional probes beyond what was in the packaged respiratory panel. I would also add that if you're doing it that at scale, we could certainly talk and add an additional target or two into the reporting.
Okay. So let's see now. So Venkatesh, I guess, or Nick, Does the panel work in other sequencing platforms, or is it specific only to Illumina sequencing?
It should work with other sequencing platforms as well. We've tested it with Illumina platform, but I don't see any restriction in being able to use other platforms as well. Yeah.
Okay. There was a question here I wanted to ask. Oh, so for Nick, how does One Codex keep the security of the data for the client?
Sure. So this is something we treat obviously with extreme care and caution. And so I should add that our entire platform is HIPAA compliant, and where it's appropriate, we are in a position where we can kind of enter into agreements with a clinical organization that might need to transmit PHI to us. So kind of security is at the utmost of our concerns around the platform. All the data that you upload to us is your data, you own it. We simply kind of help analyze it for you and on your behalf. We're not kind of taking ownership of that data as it's transferred to us. And then again, it's kind of a larger conversation for offline, but we work with a number of hospitals that we've gone through security review with and have agreements to take patient information. So that's something that we feel very good about, and obviously is kind of, we have a dedicated team that works on and maintains kind of the security of that application.
Okay. What are the sample types that you have seen research apply to the respiratory panel? For example, blood, human nasal swab, saliva, yeah, so basically what are the different sample types that you've seen people use?
Yeah, so we have seen our customers use quite a bit of different variety of sample types. We've seen nasal swabs as well as bronchoalveolar lavage fluid, as well as nasopharyngeal swabs. So there is a good variety of sample types that has been used. We've worked with some of the leading labs who kind of started working very early on in the COVID-19 pandemic, who've been using our panels and have reported excellent results. And so in these cases, the labs have used quite a variety of sample types. But essentially, those are some of the sample types that we've heard of being used.
Okay, thank you. For Nick, is One Codex available offline when you don't have an internet connection?
So it's not. It is a cloud-based system, and we've made it work on all kinds of lockdown hospital networks and Department of Energy networks and the like. And so it's fairly portable, but it does require an internet connection.
So for Venki, I guess, how much total RNA is needed to start the panel? And I think you've already answered this question. Have you seen customers using RNA from saliva samples? So I guess really ultimately, what's the input?
Yeah, so I think with the input, what we see is that about 100 nanograms of total RNA is good to start with. If you're going to multiplex heavily, about 200 nanograms should be a good starting point as well. So about somewhere in that range, about 100 to 200 should be sufficient for the panel.
Again, I think maybe for both, depending on what is the limit of detection with regards to whether it's a single infection or a mixed infection. Do you have any data with regards to that?
So yeah, like I showed through my slides, we've tested 100 copies spiked into human background RNA, and we are able to detect that quite well, even for a really small template RNA genome like the Boca virus. So I think that's kind of, in my opinion, an extreme that you'll see, and we are able to detect that quite well. Provided a larger template size, you may be able to go even smaller. Through our early access partnerships, we've seen customers run fairly challenging samples with CT values upwards of 30 and still getting pretty good co-infection detection. So yeah, I mean, I think all those ranges should be able to work fine with our panel.
And I guess kind of a related question, in real life scenarios where the CT can vary between samples, I guess number of copies, how do you suggest? Do we have any suggestions with regards to pooling samples and sequencing them so that you don't have one sample drowning out another?
Yeah. This is a fantastic question, and so we've tried to answer that question ourselves. And to be honest, I think when you're going to mix together samples that have like super varying viral loads with like something that is at a lower 20s CT value and one that is at a higher 30, you may see some interference, right? Like you will see some skewing of the samples, and so skewing of the reads going to preferentially to the ones with the higher viral loads. And so the recommendation, of course, will be that as far as possible, you should pull together samples that are at, say, the similar level of degradation or similar CT values in order to get the best performance. But again, even in the most tricky combinations, you'll probably not see more than maybe 5-fold difference between the reads going to a one sample that is at high viral load compared to the other. But of course, the recommendation would be that you pool similar CT values together as samples.
Okay. I'm sorry, Ernie.
No, please go ahead.
This is a related question that I think this kind of dovetails with on the informatics side around LOD. So I just wanted to chime in that, we showed an example here with 1 million reads, right? You don't need 1 million reads where 70% are mapping to the viral targets to make detection calls. You need a far lower number of reads, 50 to 100,000. So to the extent that you do have uneven pooling across a large number of samples, I think that as long as you're sequencing to modest depth, which is a fairly large number of samples on a MiSeq, you're not really going to run into trouble around LOD on the informatics side either.
Thank you. So for Nick, can we run One Codex as an app on DNA Nexus, or are there plans to implement it on DNA Nexus platforms as an app?
No, so it's a standalone application.
Okay. So for Venki, I guess, so does the library yield vary based on how much, if any, infection is detectable? So for example, if a sample doesn't have any virus, does it still make a library that can be sequenced?
Yeah, somewhat. I think it does depend on, it does depend on what, if any, viruses you have in the sample, definitely, yeah. Okay.
And I think we're getting close to the time here, so I'll ask one last question, if that's okay. And they all kind of sort of roll together. So basically, are there sequencing recommendations with regards to the respiratory panel? I guess how many samples you can sequence at once, that sort of thing?
Yeah, so we've tested it against, in our internal runs, what we see is that about 250,000 reads should be sufficient to ensure, really low viral copies in the samples. And so let's say, for example, you were running a MiSeq v3, which has about a 25 million read output. You can sequence up to 100 samples if you're aiming for that particular, read depth per sample. Okay. Actually, I also wanted to take one quick question here, which was around, and Ernie, I'll just quickly read this out, which is, have you tested the panel for enrichment performance on a large number of real-world wild-type samples and from different sources? And I'm asking because some strains of viruses might differ quite a bit from each other in their genomes. Can your panel enrich for any strain of the targeted virus? So this is a great question. What we see is, at least in the experiments that we've done for our app node that you'll find on our website that talks about the data that I showed today, we've of course looked at only the synthetic viruses, but we've also done a lot of and we've done some simulated, essentially we've looked at simulated data internally, and what we see is that we are able to detect viruses that have 10% mutations, random mutations that were introduced in these sequences of the viruses that we've tested against. So we have tested against 10%. So at that rate, you should be able to enrich for multiple different strains of viruses that we target through our panel. Great question.
Great. So it does look like we're coming very close to our time, and we want to be very respectful of everyone's schedule. So for any questions that we didn't get to or have the opportunity to address, please rest assured that we will reach out to you directly. And as just a quick reminder, after you leave the webinar, you will be directed to a brief survey. If you could just spare a few moments to fill it out, we would really appreciate your feedback. I'd like to thank again both our presenters, and thanks to all of you for joining us today. Until next time, remember that science doesn't stop, and neither will we. So stay safe and have a good day.